Good afternoon, students. In the last lecture, we were discussing about the selection methods in the plant breeding. Okay, there are four principal methods which are used to develop the new crop variety. Okay, the first one, 1 minute. Yes. There are four principal methods which are used to improve the crop variety. Okay, the first one, it is the domestication, plant introduction, and acclimatization. Second one is the selection, and the third one is the plant hybridization, and fourth one is the mutation breeding. Out of these four methods, we have discussed in detail the first method, domestication, plant introduction, and acclimatization in detail, and we were discussing about the different selection methods in the plant breeding. Okay, we have discussed that there are two methods of the selection. The first one is the natural selection, another one is the artificial selection. Okay, the artificial selection, it is the selection which is made by the human being for their own benefit. Okay, artificial selection, it is the selection of the plants made by the human being for their own benefit. There are three methods of the artificial selection. The first one is the mass selection, second one is the pure line selection, and the third one is the clonal selection. Okay, three methods of the artificial selection. First one mass selection, second one pure line selection, and third one is the clonal selection. Okay, we are going to discuss these three methods of the artificial selection one by one. Okay, and we were discussing about the first method of artificial selection, and this is the mass selection. Okay, mass selection means we have discussed that mass selection, it is nothing but mm, the elite group of plants, or we can say the vigorous and the healthy plants, are selected from the mixed up population on the farmer's field. Then these elite group of plants are harvested, their seeds are collected, these are preserved, these are composited together, and these are used for sowing to get the crop in the next season. Okay, and this procedure, it is repeated till the uniformity in the desired character is obtained, and later on this strain, it is released as the new variety. Okay, the process is called as the mass selection. It is considered as the one of the oldest, simplest, and easiest method of selection which is used for the crop improvement. Okay, then we have also discussed the field technique which is used for the mass selection. Okay, the mass selection is generally practiced in the cross pollinated crop, because this type of method, it is completely depending upon the genetic variability in the plants. Okay, more the genetic variability, more will be the chances of getting the new improved variety. Okay, because the selection, mass selection, it cannot be carried out in the population which is already uniform. That's why this method, it is not used for the self pollinated crops, because the self pollinated crop, these are natural homozygous, and these are having very less genetic variation. But the cross pollinated crops, these are having more genetic variation. That's why we can develop the new crop variety by using this method in the cross pollinated crop. That's why the selection method, it is generally practiced to improve the cross pollinated crops. Okay, then the process, it takes about 8 years to develop the new variety. Okay, we have discussed in detail all the activities which are used in the first year, second year, then third to fifth year, then sixth to eighth year, and lastly we have discussed in the eighth year the strain which is performed better than that of the existing superior variety is released as a new variety. Okay, we have discussed in detail the field technique which is used for the mass selection. Okay, we were about to discuss the two methods of the mass selection. Okay, in this one, the first method is called as the Halides method. Okay, Halides method, it is the method which is based on the Lamarck's doctrine, which is which is very famous, and according to this doctrine, acquired characters are inherited. Okay, acquired characters are inherited. In this method, the crops, these are grown under the best environmental conditions. Okay, crops are grown under the best environmental conditions means what? That is, the crops are provided with the maximum amount of fertilizers, then proper irrigation is given, and then later on from this population the mass selection is carried out. Okay, understand that in method, the crops, these are grown under the best environmental conditions. These crops are provided with the maximum amount of fertilizer and irrigation facilities, and later on the mass selection is carried out from this population. Okay, and thus this method is based doctrine, acquired characters are inherited. ओके, लक्षात आलं? अक्वायर्ड कॅरेक्टर्स आर इनहेरिटेड म्हणजे काय? तर प्लांट्स हे सगळ्यात चांगल्या बेस्ट एन्व्हायरमेंटल कंडिशनमध्ये ग्रोन केले जातात. त्यांना खूप सारे फर्टिलायझर्स प्रोव्हाइड केले जातात, त्यांना व्यवस्थित वॉटरिंग केलं जातं. त्यामुळे काय होऊन जातं? त्यांची ग्रोथ व्यवस्थित होते. ओके, म्हणजे हे कॅरेक्टर्स काय आहेत? अक्वायर्ड आहेत. Okay, that is due to the surrounding environmental conditions, but generally the acquired characters are not inherited. ओके, म्हणजे त्यांची ग्रोथ जरी व्यवस्थित झाली असली, सगळी झाली असलं तरी काय, हे कॅरेक्टर्स हे इनहेरिट होत नसतात. ओके, दॅट्स वाय दिस मेथड इज नॉट प्रॅक्टिकल. Okay, it is not practical that all the best environmental conditions are provided to that plant, and from these group of plants the mass selection it is carried out. Okay, but this is not practical, that is acquired characters are not inherited, and that's why the another method was introduced. 1 minute please. Another method was introduced, sorry, another method was introduced, and this method is known as the Rimpau method. As per this method, the crops, these are grown under the ordinary or even unfavorable environmental condition. Okay, opposite to Hallett's method, in the Rimpau's method, the crops, these are grown under the ordinary or even under the unfavorable environmental conditions. Unfavorable environmental conditions or ordinary condition, we can say the minimum amount of water or the fertilizers are provided to the crops. Okay, minimum amount of water and fertilizers are provided to the crops, and the plants looking better, these are selected. Okay, and the Rimpau's method, it is more acceptable method. It is more acceptable method because it is easily applicable. Okay, it is easily applicable. And secondly, the environmental variability which influences greatly the characters like the yield, these are considerably reduced. Okay, environmental variability which influence characters like the yield, these are reduced, and the genetic variability is allowed to full expression. Okay, this Rimpau method, it is more acceptable method because there are two reasons. The first reason, that it can be easily applicable or it can be easily used. Okay, that is the first reason. And the second reason is that the environmental variability which influences the different characters like the yield is greatly reduced, and the genetic variability is allowed to full expression. Okay, that means the characters, to the genetic variability, the characters can be transferred from the one generation to the another generation. Okay, but the variation which is created, or the variation which is influenced by the surrounding environmental condition, it cannot be transferred from the one generation to the another generation. That is the reason the Rimpau method it is more acceptable. Okay. Okay, that is all about the two methods which are used in the mass selection. The first one is the Halides method, and another one is the Rimpau's method. Okay, then another important thing that from studying the first the all the technique, all the field technique in the mass selection, you can be easily observe that the mass selection it is entirely dependent on the phenotypic characters. Phenotypic characters, or we can say the morphological characters, which can be easily observed with the naked eyes. Okay, the mass selection method it is entirely dependent on the observation of the phenotypic character, or we can say the external characters of the plants which can be easily observed with the naked eyes. Okay, and that's why we can say the mass selection it is more art than science. Okay, it is more art than science. And the success of mass selection method it is depend on the extent up to which the phenotypes are the actual expression of the genotype. Okay, the mass selection method it is success depends on the extent up to which the phenotypes or the external characters are the actual expression of the genotype. म्हणजे जे काही महत्वाचे कॅरेक्टर प्लांट्स मध्ये आहेत, हे कॅरेक्टर हे एन्व्हायरमेंटल कंडिशन मुळे क्रिएट नाही झालेले पाहिजे. तर ते कॅरेक्टर कशामुळे असले पाहिजे, तर ते त्याच्यामध्ये असलेल्या जीन्समुळे असले पाहिजे. तरच काय होईल, ते कॅरेक्टर एका जनरेशन मधून दुसऱ्या जनरेशन मध्ये पास होतील. जर त्याच्यामध्ये कॅरेक्टरमध्ये येणारं व्हेरिएशन हे एन्व्हायरमेंटल कंडिशनमुळे असेल, तर ते कॅरेक्टर्स एका जनरेशन मधून दुसऱ्या जनरेशन मध्ये पास होत नाहीत. Okay, that is all about the mass selection. Then in this slide you can see the application and the uses of the mass selection. Okay, the first point here you can see the mass selection it does not create any change in the genetic constitution or genotype. It only sort out the different types of the genotypes which already exist in the population. Okay, that means the mass selection it does not create any new character which is not present in the population. Okay, not creating any new genes. Okay, but it only sort out the different types of the genotypes which already exist in the population. ओके, लक्षात आलं. म्हणजे याच्यामध्ये कुठल्याही नवीन प्रकारचा जीन प्रोड्यूस होत नाही किंवा कुठल्याही नवीन प्रकारचं कॅरेक्टर हे तयार होत नाही. याच्यामध्ये काय होतं, फक्त मिक्स्ड पॉप्युलेशन मध्ये जे चांगले कॅरेक्टर्स आहेत फक्त आपण काय करतोय तर त्याचं आपण सॉर्टिंग करत असतो. फक्त चांगल्या कॅरेक्टर्सचं सॉर्टिंग करतो. कुठलंही नवीन कॅरेक्टरचे पॉप्युलेशन मध्ये प्रेझेंट नाही हे त्याच्यामध्ये क्रिएट होत नसतं. Okay, that is the most important thing while studying the mass selection. It does not create any change in the genotype. It only sort out different types of genotypes which already exist in the population. Okay, then here the method it is best applied to the cross pollinated crops. Okay, we have discussed it just in the lecture, that is the this method is generally used for the improvement for the cross pollinated crop. Okay, because the mass selection, the success of the mass selection it is depending upon the genetic variability of the crops. More the genetic variability, more will be the successful, it will be successful to develop variety by using the mass selection. Okay, and as the cross pollinated crops are more heterozygous and natural the genetic variation, this method is used for the development of new crop variety in the cross pollinated crops. Okay, this is the method which is best applied to the cross pollinated crops for the production of new variety, then purification of unimproved or mixed varieties, and the third one maintaining purity of variety where supply of seed not properly organized. Okay, this method is generally used in the cross pollinated crops for number one production of new variety, Number two, purification of unimproved or mixed varieties. And third one, maintaining purity of variety where supply of seed not properly organized. Then here the advantages of the mass selection. Okay, advantages of the mass selection. Advantages, first one, it is easiest, simplest and quickest method of crop improvement. Okay, advantages, it is easiest, simplest and quickest method of crop improvement in the cross pollinated crop, because in this method just we have the field technique, there is a no testing of variety. Okay, in the 8th year the new variety it is produced, and that variety it is not tested for number of generation. Okay, that's why it is easiest and simplest. And the another reason is that there is no control on pollination. Okay, you don't have to control the pollination in this method, because generally it is practiced in the cross pollinated crops. Okay, there is no control on pollination. When you think in other methods which are used for the improvement, for example in pure line selection, in that is the second method of selection, where the control on pollination is the most important factor. You have to control the pollination, but this is the method which is very simple. Here there is no control on pollination. ओके, म्हणजे नॅचरली क्रॉस पॉलिनेटेड होतात, याच्यामध्ये तुम्हाला कुठल्याही प्रकारचं पॉलिनेशन कंट्रोल करायची गरज नाही. परंतु प्युअर लाईन सिलेक्शन जी सेकंड मेथड आहे त्याच्यामध्ये मात्र पॉलिनेशन वरती तुम्हाला कंट्रोल ठेवावा लागतो. त्याच्यामुळे काय, ह्याच्यापेक्षा ती मेथड थोडीशी अवघड आहे. Okay, that's why here we are saying the mass selection, it is the easiest, simplest and the quickest method. Mass selection, it is easiest, simplest and quickest method because there is no testing of variety. At the end of 8th year the variety is released without any testing. And second important factor is that there is no control pollination. Okay, then the second advantage of using this method, it is the only method for improving the local or the wild varieties to meet the need of the farmers. Okay, it is the only method for improving wild and local varieties to meet the immediate need of the farmers. Okay, then the third one, the this method, it is the basis of all crop improvement program. It is the basis of all crop improvement programs. After this, the other methods can be exercised for the further improvement. ओके, म्हणजे जेव्हा तुम्हाला क्रॉस पॉलिनेटेड व्हरायटीमध्ये नवीन क्रॉस पॉलिनेटेड क्रॉपमध्ये नवीन व्हरायटी रिलीज करायची आहे, तेव्हा सगळ्यात पहिल्यांदा तुम्हाला कोणती मेथड यूज करावी लागते, तर मास सिलेक्शन. बाकीच्या ह्या मेथड ह्या तुम्हाला नंतर यूज कराव्या लागतात. ओके, म्हणजे लगेच तुम्ही बाकीच्या मेथड यूज करायला चालू केलं असं होऊ शकत नाही. क्रॉस पॉलिनेटेड क्रॉपच्या इम्प्रुव्हमेंटसाठी सगळ्यात पहिल्यांदा मास सिलेक्शन ही मेथड यूज केली जाते. आणि जेव्हा त्याचं जेनेटिक व्हेरिएशन कमी होऊन जातं क्रॉस पॉलिनेटेड क्रॉपमध्ये, तेव्हा ह्या दुसऱ्या मेथड त्याला ऍप्लिकेबल असतात. Okay, that's why we are saying that is the mass selection method is basis of all crop improvement program, and after this the other methods can be exercised for the further improvement. Okay, these are the three most important advantages of the selection. Okay, I am repeating this one, it is easiest and simplest and quickest method because the testing of variety it is not carried out. And another important factor is that there is no control on pollination. Okay, second one, it is the only method for improving the local or wild varieties to meet the need of the farmers. Okay, and the last one, it is the basis of all crop improvement program. After this, other methods can be exercised for the further improvement. Okay, that means for the development of new variety in the cross pollinated crop, firstly the mass selection method is used. And when the genetic variation in the cross pollinated crop due to the mass selection is exhausted, later on the other methods are used for the improvement of the cross pollinated crops. Okay, these are the three most important advantages of the mass selection. Okay, now we are moving to discuss the limitations of the mass selection. Limitations, or in other word we can say the disadvantages, or the demerits of this method. Okay, the first limitation of this method is improvement. The improvement which is procured in the variety is short lived, and the anticipated results not obtained. Okay, this is the first limitation of this method. Improvement which is procured in the variety is short lived, and the anticipated results are not obtained. Anticipated results म्हणजे काय? जे आपल्याला हवे आहेत असे रिझल्ट आपल्याला एकदम 100% भेटू शकत नाही. आणि इम्प्रूव्हमेंट जी आहे, म्हणजे ज्या क्रॉपमध्ये जे कॅरेक्टर्स त्याच्यामध्ये इम्प्रूव्ह झालेले आहेत, ते कॅरेक्टर्स हे शॉर्ट लिव्ड असतात. Okay, that means whenever you are cultivating that variety for the number of generation, that number of generation, the characters, added characters in the variety get deteriorated. Yes, what is the reason why the improvement is short lived, and why the desired characters in the released variety deteriorated when the variety is cultivated for the number of generation? Okay, because in this one, the first important factor, as there is no control on pollination, there is no control on pollination, and the cross pollinated crops are naturally heterozygous. Okay, naturally heterozygous, and due to the segregation of characters, due to the segregation of characters in each generations, which deteriorates the variety, or which deteriorates the desired characters. ओके, लक्षात आलं की इम्प्रूव्हमेंट हे शॉर्ट लिव्ड का असतात? कारण आपण हे यूज कशामध्ये करत असतो? क्रॉस पॉलिनेटेड मध्ये हे यूज करत असतो. म्हणजे जी व्हरायटी रिलीज झालेली आहे ती क्रॉस पॉलिनेटेड क्रॉप्स काय नॅचरली heterozygous असतात. क्रॉस पॉलिनेटेड क्रॉप्स आर नॅचरली heterozygous. Self pollinated crops are naturally homozygous. Okay, don't forget, that is the important factor while studying the plant breeding. Cross pollinated crops, these are always naturally heterozygous, and the self pollinated crops are naturally homozygous. Okay, heterozygous means more genetic variability. Okay, and due to which the in each of the generation, due to the genetic recombinations, the genetic recombinations new due to the genetic recombination and the segregation of characters, the characters get deteriorated in the variety. ओके, लक्षात आलं. क्रॉस पॉलिनेटेड क्रॉप्स हे हेटरोझायगस असतात. हेटरोझायगस असल्यामुळे काय, तर प्रत्येक जनरेशनमध्ये सॅग्रिगेशन ऑफ कॅरेक्टर, यू हॅव स्टडीड द मेंडेल. ओके, द रूल ऑफ सॅग्रिगेशन. ओके, द लॉ ऑफ सॅग्रिगेशन. इन ईच ऑफ द generation, the characters get segregated or get separated from each other, and due to which the variety get deteriorated. म्हणजे जी व्हरायटी तुम्हाला हवी आहे ती नंबर ऑफ जनरेशन नंतर काय होऊन जाते, तर त्याच्यातले कॅरेक्टर्स हे सॅग्रिगेट होतात, सेपरेट होतात, आणि नंतर ती व्हरायटी ही डेटेरिओरेट होत असते. Okay, that is the most important limitation of the mass selection. Okay, important, because the improvement which is procured in the variety is short lived, and anticipated results are not obtained. Okay, because the cross pollinated crops, these are heterozygous, and as there is no control pollination, it causes the more heterozygosity in the variety, and due to which when this variety it is cultivated for the number of generation, due to the segregation of characters, the variety get deteriorated. This is the important limitation of this method. Okay, then the second one, this method it is not good for increasing yield. Okay, this method is not good for increasing yield. We are saying this because there are more reasons. Like the first reason is that the attention in this method, it is only given to the maternal characters. Okay, the paternal characters, these are completely neglected. The attention, it is given only to the maternal character. Okay, we are discussing the reasons for the method, the mass selection method not good for increasing yield. There are various methods in which the first reason is the attention, it is given only to the maternal character. Okay, attention is only given to the maternal character. Then the second reason is that the yield fluctuate by the environmental variation. Okay, the yield, it is generally related with the environmental conditions, and due to changing environmental conditions, the yield also get fluctuated. Okay, जो yield आहे क्रॉपचा हा जनरली कशाला कोण करत असतं, तर एन्व्हायरमेंटल कंडिशन्स, त्याच्यामुळे काय होतं, हे कॅरेक्टर सारखं चेंज होत राहतं. And thus the phenotypic characters and the environmental cannot be separated. Okay, that means the we cannot distinguish the characters owing to the environmental conditions and the characters which are owing to the genetic constitution. ओके, म्हणजे आपण असं समजा एखादं प्लांट बघितलं तर आपण असं म्हणू शकत नाही की बाबा हे जेनेटिक कॅरेक्टर्समुळे याचा इल्ड वाढला किंवा एखादं कॅरेक्टर चांगलं झालंय. असंही असू शकतं की बाबा ते कॅरेक्टर कशामुळे इम्प्रूव्ह झालेलं असेल, तर सराउंडिंग एन्व्हायरमेंट कंडिशनमुळे ते कॅरेक्टर इम्प्रूव्ह झालेलं असू शकतं. ओके, त्याच्यामुळे आपण काय म्हणतोय की वी कॅन नॉट डिस्टिंग्विश द कॅरेक्टर विच इज ओईंग we cannot distinguish the phenotypic characters which are owing to the environmental variation or owing to the genetic constitution. Okay, that is the another reason that method is not good for increasing yield. We cannot distinguish the characters owing to the environmental variability and the genetic constitution. Okay, then another reason it is not good for increasing yield is there is no control on pollination. Okay, there is no control on pollination. That's why the selected plants may get pollinated by the superior or the inferior pollen grains. Okay, these are the three reasons. That's why this method is not considered for the not considered good for increasing yield. Okay, three reasons. Repeating these three reasons again. The first one, the attention, it is given only to the maternal characters. Okay. Second one, yield fluctuates by the environmental variation, and we cannot distinguish the phenotypic characters owing to the environmental variation and the genetic constitution. And the third reason is there is no control on pollination, and that's why the plants which are selected in this method may get pollinated by the superior or the inferior pollens. Okay, this is the limitation. And third one, the method, it is not applicable for the self pollinated crops. Okay, the method, it is not applicable for the self pollinated crops because this method, it is depending upon the genetic variability within the population. More the genetic variation, this method, it can be easily used. Okay, the self pollinated crops, just we have discussed, these are homozygous. That means these are not having more genetic variation. Okay, and the cross pollinated crops, these are heterozygous, and these are naturally genetic variable, and that's why this method is applicable only for the improvement of the cross pollinated crop, and this method cannot be used for the improvement of the self pollinated crops. Okay, these are the limitations of the mass selection, though there are three limitations for using this method, but this method, it is very popular in farmers. Okay, though there are certain type of limitation, this method is very popular among the farmers. And all cultivated crops and their local strains, these are the result of mass selection. Okay, all the cultivated crops and the their local strain, these are the result of mass selection. लक्षात आलं, जरी ह्या मेथडला काही लिमिटेशन्स जरी असले तरी ही मेथड फार्मर्स मध्ये खूप पॉप्युलर आहे. ओके, किंवा कारण का, because the farmer wish to improve, this method can be used by the farmers who wish to improve their agricultural crops, but they does not have any knowledge regarding the inheritance of character. ओके, फक्त सिंपल आहे. याच्यामध्ये काय करायचं आहे, तर मिक्स्ड पॉप्युलेशन मधून फक्त जे चांगल्या कॅरेक्टर्सचे व्हिगरस हेल्दी प्लांट्स आहेत, फक्त त्याचं डिझायर्ड कॅरेक्टरचे ते प्लांट तुम्हाला सिलेक्ट करायचे, त्यांचे सीड्स हार्वेस्टिंग नंतर कलेक्ट करून प्रिझर्व करायचे आणि ते नेक्स्ट जनरेशन घेण्यासाठी ते यूज करायचे. असं वर्षानुवर्ष केल्यानंतर त्या व्हरायटीच्या त्या क्रॉप सिलेक्टेड प्लांटमध्ये मध्ये एक युनिफॉर्मिटी येते आणि तीच नवीन व्हरायटी म्हणून रिलीज केली जाते. ओके, आणि जेवढे काही लोकल स्ट्रेन्स असतात, म्हणजे जे लोकलला आपण जे समजा एखाद्या शेतकऱ्याकडून लावण्यासाठी काही घेत असतो बियाणं, तर ते काय, ते लोकल स्ट्रेन असतात. हे सर्व जे लोकल स्ट्रेन आहेत हे कोणत्या मेथडने डेव्हलप झालेले असतात, तर हे मास सिलेक्शन मेथडने मास सिलेक्शन मेथडने ते तयार केलेले असतात. That's why we are saying, though there are certain type of limitations for using this method is very popular among the farmers, and all the cultivated crops and their local strain, these are developed by using this method. Okay, these are the limitations of the this method. Okay, I think this is enough for today. We have discussed in today's lecture the two methods of the mass selection, and also we have discussed the advantages of the mass selection and the limitations of the mass selection. Okay, in the tomorrow lecture, we will be discussing the second method of the selection, and that is the pure line selection. Okay, this is enough for today. माझा आवाज